Okay, now we are going to change gears a bit here because up until now we've really been talking about what the nervous system is. So we've talked about neurons, we've talked about action potentials, we've talked about synapses and synaptic transmission and neurotransmitters, and we talked about the anatomy of the nervous system, the different parts of the nervous system. So we're going to move from that to talking about what the nervous system does. And that's actually what we're going to be doing for pretty much the rest of this class. So the good news is, though, that the nervous system actually only really does two things. One is taking information from outside the body, or sometimes inside the body, and sending it to the brain, and then reacting to that information in the form of movement. So the first part of that, we're going to be talking about the sensory systems, and then later on we'll talk about the motor systems. So we'll go through the different sensory systems in order. The first one is going to be the chemical senses, and we'll talk about what that means in a minute. But first, before we do that, let's talk about just some general principles dealing with the sensory system. So first of all, when we're dealing with sensory systems, the information that the sensory system deals with is called a sensory stimulus. So whatever the information is that a sensory system gets is the stimulus. So in the case of the chemical senses, for example, the two types of stimuli, or two sensory systems that deal with chemical stimuli, are taste and smell. So that's what we're going to deal with in this chapter. So those are sensory systems that use chemicals as a stimulus. And then the mechanical sensory systems, or the sensory systems that detect mechanical stimuli, and that means like actual physical force or deflection against a sensory surface, are your sense of touch. Sound we consider a mechanical sense because, as we'll learn, sound is just the movement of air molecules, and then movement of the head or the body itself. And then electromagnetic fields can also be a sensory stimulus. Now, the only kind of electromagnetic stimuli that we are able to detect is light, and so that's through the visual system. And then there are other animals that can detect electric fields and magnetic fields, but for humans, that's not really a stimulus that we, and most other mammals, in fact, that's not really a stimulus that we normally are able to detect. So that's what we're going to be talking about. This chapter will deal with the chemical stimuli, and then we're going to talk about the somatosensory system and the auditory system later, and then we'll also be talking about the visual system as we go along. Now, a stimulus will have several properties, and the job of the sensory system is to detect and encode or interpret those properties. So the first thing that a stimulus has, that the sensory system has to be able to determine, is the modality, which basically just means what kind of stimulus is it? Is it it has to be able to tell the difference between a mechanical stimulus and a chemical stimulus, and so on. Now, typically, modality is determined by or is encoded by the type of sensory receptor. So every sensory system has a receptor, which means a cell that detects the sensory stimulus. So in the case of, for example, the visual system, we'll learn later that there are photoreceptors in the retina. That means cells in the retina that respond to light. In fact, usually the sensory cell or receptor will convert the sensory stimulus into some kind of electrical signal that then gets converted or gets sent to the brain. Now, the modality, again, is determined by the type of receptor, and most receptors are only tuned to detect one type of sensory stimulus. So, for example, the light receptors in your retina are only really geared toward detecting light, but they can be activated in other ways. So, for example, if you've ever, and you can do this yourself, if you've ever closed your eye and kind of rubbed your eyeball or kind of pushed against the side of your eyeball with your finger, what you may notice is a little blob of light that appears in your visual field. Now it actually appears on the opposite side of your visual field of where your finger is, but that's because of the fact that your eye flips information that comes into it. We'll explain what that is later. But what's happening there is you are pushing against your retina and you're physically deflecting it, and that motion does actually cause activation of the photoreceptors or the other neurons that are connected to the photoreceptors, and that signal gets sent to the brain. But your brain doesn't detect it as a physical force, although you also have touch receptors in your eyelid and the eyeball itself that detect the physical movement or the physical stimulus, mechanical stimulus, I should call it. But the photoreceptors don't send a signal to the brain that says I'm being physically deflected. There's a mechanical stimulus being applied to me. Instead, it gets reported to your brain as light. So that's why, even though you're not seeing any actual light, your photoreceptors are reporting to your brain that there is a little ball of light that is coming somewhere in your visual field. So again, each receptor and each sensory system is able to detect and interpret usually just one type of sensory stimulus. The next piece of information that you would want to know about a sensory stimulus is its intensity, which basically just means how strong the stimulus is. Now, that depends on the modality. So in the case of a chemical stimulus, for example, the intensity really is just the concentration, how much of that chemical is present. With mechanical stimuli, usually you're talking about the amount of force being applied to the sensory receptor. And then with light, you're talking about the brightness of the light. So in each case, the sensory system has to be able to determine that, how much, what the intensity of the stimulus is. And then location usually means where did the sensory stimulus come from. Now, in some sensory systems, that's pretty straightforward. So with the visual system, for example, location just means where in your visual field did a given photon of light come from, or where is an image coming from in your visual field. With sound, you may also want to know where did a sound come from with respect to the position of your ears. And in touch is pretty straightforward too. If you're talking about where did a touch stimulus come from, you really mean where on your body or on your skin was a touch or mechanical stimulus applied. Whereas with the chemical stimuli, chemical sensory systems, location is a little more complicated, because in the case of taste, for example, as we'll learn later, your tongue is the only place where you really taste things, and there is no really difference in location across the tongue that tells us much. Whereas in smell, again, you might be able to determine where a scent is coming from. And so again, your sensory system has to be able to determine that also. And then the last thing that your sensory system has to be able to encode is the timing of the stimulus, which usually just means when did the stimulus occur, and when did it stop, and how long did it last, or the duration. And typically this is pretty simple to encode. It's just when did the receptors and the neurons connected to those receptors become active, and when do they stop being active. But there are some specialized sensory systems that can encode timing of a stimulus by different frequencies or different sort of on-off patterns. But we'll see some examples of that later on. Now, getting specifically to the chemical senses, there are only really two that we're going to talk about. So taste is probably the simplest of the sensory systems that we have, and the long name for taste is gustation. But usually it means the detection of water-soluble chemicals. Aqueous is just a word that means water-soluble, that come in contact specifically with the tongue. So typically this means things that are chemicals from substances that get into your mouth. So it's it's basically a contact sensation. You're not able to really detect chemicals with your tongue at a distance. So anything that physically comes in contact with your tongue, if it contains water-soluble molecules that have receptors on the tongue, then you will be able to detect that. Now, obviously, and we'll talk about kind of how each of these sensory systems possibly evolved. So in the case of taste, this is probably the oldest and most conserved sensory system, at least among vertebrates, possibly among all organisms, because even bacteria, even single-celled organisms, have the ability to detect chemicals in their environment, in the water that they live in, and are able to respond to that. So probably taste is one of the oldest sensory systems to have evolved, and that makes sense because taste is, again, a sensation associated with things in your mouth, which of course is where food goes. So, and in fact, all animals pretty much have some sort of taste or taste-like sensory system that's associated with receptors in the mouth or whatever part of the body food goes into. And so that's probably the main purpose of taste, is for you to determine whether the substance that you've placed in your mouth is food or not. And so, as we'll see, most of the things your gustatory system detects are linked to food. Olfaction, meanwhile, is the fancy word for smell. So smell, we're still in one of the chemical sensory systems, so the detection of chemicals, but instead of water soluble, the olfactory system detects volatile chemicals. So these are chemicals that, instead of being dissolved in water, are essentially dissolved in air. So that can be food, right? So some food releases molecules into the air through just sublimation or evaporation, and then your olfactory system can detect those and determine if something is edible or not. But of course, other things emit volatile chemicals, so your sense of smell can also be used to perhaps detect danger. So a predator, for example, might give off molecules through sweat, for example, that you might want to be able to be aware of. Other things that might be poisonous or toxic might have their own scent associated with them. Things like fire, of course, have an odor. So these are all things that, as an animal, you might want to be aware of, so that you can avoid, in addition to things you might want to eat or be attracted to. Also, sense of smell can be used to identify other members of your species. So olfaction can be important for detecting potential mates, for example, or being able to tell family members from other members of your species that you're not related to. So there are all kinds of reasons why that sense of smell is important. Another big difference between taste and smell, again, taste is only useful for things in your mouth. So that's the source of your sense of taste, whereas your sense of smell can be used to detect things at a distance, because anything that has a smell, by definition, is emitting molecules into the air, and depending on how far those molecules travel, you can detect those molecules at a long distance, and you can even use your sense of smell to locate objects. Now, these are the two chemical sensory systems that we're going to talk the most about, but there is at least one more. So there are also chemoreceptors or cells in your body that can detect chemicals inside your body. So that would mean chemicals in your bloodstream, for example, that can tell your brain about the internal state of your body. So, for example, you have pH receptors in your bloodstream. Actually, they're in a part of the body called part of the brain called the hypothalamus. And those are important because changes in pH usually are reflective of things like the internal body state, the pH state of your body tissues, and then there are also detectors for oxygen, for example. And so those are chemoreceptors in the sense that you're detecting chemicals. The big difference, of course, when we're talking about those kind of chemoreceptors is that you're not usually consciously aware of those kinds of sensations. And they're important because they help regulate, so it's a way for your brain to regulate things like oxygen levels and pH. But again, you're not usually you don't have conscious access to those things. So we won't be talking about those very much, at least in this chapter. We'll come back and talk about the hypothalamus more when we get to the autonomic nervous system. So let's focus now on taste. So again, taste is about detecting chemicals that are water soluble that are in your mouth. And taste, again, is probably the simplest of the sensory systems, because there are only really 5 sensations associated with taste. And they are associated with 5 different sort of categories of molecules. So the sensation of sweet, for example, is usually associated with molecules like sucrose, glucose. These are collectively called called sugars. Or sometimes called carbohydrates. So these molecules are important because they're actually made primarily by plants. So plants produce carbohydrates by taking carbon dioxide from the air and water and making those molecules, sugar molecules, using energy from the sun. And plants use them as a way to store energy. So they can then break down sugars later when their cells need energy. And so and that's called photosynthesis, whereas those of us that eat plants, and so most animals, can then use that energy also by breaking those sugar molecules down as well. And so sugars are an important source of energy that we get from plants. So that's probably why we have a sensory system that can detect sugars specifically. Because again, we can use sugars for energy. It's also probably why, and we'll come back to kind of the emotional content of these flavors in a minute. Salty is, so we talked about sweet. So salty is a sensation that's associated with salt, but what we really mean by that is solutions of sodium ions. So sodium specifically, and sometimes other monovalent cations like potassium actually will elicit the sensation of saltiness. And in fact, when we put salt on food, so the shakers of salt that you would have on your table, those are of course usually just crystals of sodium chloride. And you know from chemistry that if you take sodium chloride crystals and dissolve them in water, they dissociate into the sodium ions and the chloride ions. And it turns out that when you're tasting salt, what you're really tasting is the sodium ions. And we have a sensory system for that because salt is these ions. We've already seen plenty of examples why by maintaining the ion concentration in your body is important. So it's important for neurons, of course, because maintaining the extracellular concentration of sodium and potassium and chloride and all these ions is important. But all your cells, in fact, need to operate within a narrow range of salt concentration or ion concentration. We call this homeostasis. So for anything that any parameter like salt concentration, for example, that has to be kept within a narrow range, the process of keeping your cells in that range is called homeostasis. So we need to be able to detect salt in our food in order to maintain that homeostasis, because you also lose salt whenever you lose water from your body because it's water soluble. So whenever you lose water through sweat or urine or whatever, or from bleeding, for example, you are losing salt, and so the ability to replace that salt is important. Sensation of sour, meanwhile, is associated with anything that has a low pH, which of course means it has a high concentration of protons. And that's important because, again, just like with sodium and other ions, the pH of your body, bodily fluids and blood, has to stay within a narrow range for your cells to to operate. And so your brain needs a way to tell if the food you're eating has a high or low pH in order to maintain that homeostasis. Bitter is a little more complicated because there actually isn't a single canonical bitter molecule. There are actually a number of different molecules that elicit bitter flavor. The quinines, for example, are often the typical bitter compound, but there are lots of others. And the things that elicit a bitter sensation are usually large, complicated molecules, and they don't necessarily have a lot in common, except that many of the molecules that we associate or perceive as bitter are either themselves toxic or poisonous, or are found in plants or other sources that contain toxins. So probably that sensation evolved to be able to detect and avoid things that have poisonous or toxic substances in them. And then umami, meanwhile, is the sensation that you get from consuming glutamic acid, or in either the acid form or the free base form, which is called glutamate. And this is usually associated with foods that have a high protein content, because glutamate is one of the amino acids that you can use to make proteins. And so presumably that sensation of being able to taste glutamate is useful because it tells you if the food you're eating has protein in it, because of course you need protein in your food in order for your cells to break down those proteins and use the amino acids to make new proteins, and to stay alive. The word umami, by the way, is translates from I think it's Japanese, to delicious or savory, and in fact sometimes just savory is what the word that we use to describe umami. So anything with kind of a meaty or savory flavor will have a high glutamate concentration, and therefore will have that umami flavor. By the way, the free base form of glutamic acid is glutamate, and then you can get concentrated glutamic acid or glutamate, but you have to get it in its sodium salt form. And so, and glutamate, you probably know, when it's in its free base, has a charge of -1. So sodium has a charge of +1. So usually it's a atom of sodium with a molecule of glutamate, and then often this is called, when you buy just bottles of it off the shelf, as monosodium glutamate. So one atom of sodium, one molecule of glutamate, or it's just abbreviated MSG. So MSG is just the sodium salt of glutamate, and it's used as a flavoring or it's used to add umami flavoring to food, just like sodium chloride is used to flavor or give food a salty flavor, or again anything with an acid in it, like lemon juice or vinegar, is used to make things more sour. MSG unfortunately gets kind of a bad rap because it used to be associated, or it was thought that foods that were high in MSG were bad for you, bad for your cardiovascular system, and that's not really the case. Anything with MSG in it is perfectly safe. The only reason to maybe avoid MSG is because anything with a lot of MSG added, if it's supposed to be food that has a lot of meat in it, so meat obviously usually has a lot of protein, but if someone has added a bunch of MSG to, say, a soup or something, that's usually to mask the fact that it has very little flavor or not a lot of actual meat to it. And actually, that brings up the other interesting thing about taste, is that some of these tastes or some of these sensations we think of as pleasant, and others we think of as unpleasant. And whether something is pleasant or unpleasant can kind of depend on the context. Now, we're not going to get into how we perceive the emotional content of food or anything else really, but you can kind of, again, based on what these molecules do and how they're used in the body, you can kind of predict how they're going to be perceived emotionally. So sugar or the sensation of sweet is usually a positive sensation. So we usually enjoy food that is sweet. And that's because you pretty much always need more energy. So your cells are always using energy. Unless you've really just gorged yourself, there's really no such thing as too much energy available. So that's why the probably the sensation of sweet always is evokes sort of positive emotions. Saltiness and sour both actually, meanwhile, can be either positive or negative. So some food can be too salty or too bland. Some things can be too sour, but some foods are actually taste better if they're sour. And again, that reflects the fact that those two parameters have to stay within a narrow homeostasis range. So anything too salty will taste bad because that excess salt will drive or has the potential to drive your bodily fluids outside their homeostasis. Same way with pH. Whereas if you're low on salt, anything that tastes salty will taste pleasurable. Bitter, meanwhile, is almost always negative. We almost always avoid bitter flavors because, again, typically, or the most common bitter flavors are toxic. But again, not all things that we perceive as bitter are bad for us. And in fact, some things like coffee, for example, have bitter flavors in them that can actually be pleasurable. So if you enjoy coffee or some certain herbs or vegetables that have bitter compounds in them, again, it's not because those are toxic. It's just that they happen to activate the bitter receptors on your tongue. But because they're usually combined with other more pleasant flavors, we have sort of an emotional connection, a positive emotional connection to those. So that's another thing to point out is there's the pure sensation of each of these tastes, but then there's also an emotional component to each one, which is not necessarily connected to the sensation directly. Same way with umami. So again, umami is generally always a pleasant sensation because, again, your cells always need more protein. All right, so again, those are the sensations associated with taste. So the sensory surface associated with taste, or the gustatory system, is the tongue. So the tongue is, for us, is basically just a big muscle that sits in the floor of your mouth, attached to usually the floor of your mouth and the back of your pharynx. The pharynx is basically just the cavity that connects the mouth with the nasal cavity. So, of course, food and other substances that you eat or drink go in through the mouth and kind of wash over the tongue. And so on the surface of the tongue, you have all these little bumps and cavities. The bumps are called papillae. So a single papilla is just a bump on your tongue. And there's different kinds of papillae, depending on which part of the tongue you're looking at. Now, it used to be the case if you learned about the sensory systems, probably like 10 years ago, you might have learned that different parts of the tongue are attuned to different tastes. I think they used to say that like the tip of the tongue is more attuned to sweet flavors. That's not really the case. The taste receptors are for most people are pretty much evenly distributed around the tongue. So the different papillae all have the same kinds of receptors. They only really differ in their shape. But what's really important about the papillae is that if you look at the zoom in on one, you find that each of those little bumps is surrounded by, of course, covered in saliva. And then the inner lining of each papillae is covered by these clusters of cells called taste buds. So the taste buds are lining those papillae along the side. And each taste bud is really consists of a little hole in the surface of the tongue called the taste pore. And then that leads into a cavity that contains the actual taste cells. So the taste cells, which are these kind of pink things here, are the cells that are actually going to detect the tasteds or the molecules that you can taste. So these taste buds, again, they line all these papilla, and of course, everything is covered in saliva. So your mouth cavity at all times is covered in an aqueous solution, which is saliva, which is secreted by glands, saliva glands that are under the tongue and other parts of the mouth. So at all times, there's an aqueous solution coating these cells. So when you eat food or drink something, the taste molecules associated with that food, as you chew it up or as you put it on your tongue, will dissolve in the saliva and make their way down into the grooves around these papillae and then enter the taste pore, and then that's where they interact with the taste cells. So if we look up close at one of these taste buds, this is what it looks like. So again, each one is surrounded by the lingual epithelium. So that just means epithelial cells. Epithelial cells are just the cells that line your mouth. Anytime you have a tissue that separates one part of the body from another, it's usually an epithelial layer. And then again, the gaps in that lingual epithelium form the taste pores. The taste receptors actually have little tiny cilia that stick up into the taste pore, which is where some of the taste receptor proteins are found. And then the taste receptor cells kind of take up this whole area. This is one thing. Well, we'll come back to that in a minute. So you also have inside these the taste bud these basal cells. They provide some support to the taste receptor cells. They also have the ability to some of them to turn into taste receptor cells. So in fact, it is possible to destroy taste receptor cells. So if you, for example, put something really hot in your mouth, or something that's has any sort of toxic substance or has very high concentration of things like acid or salt, you can kill these taste receptor cells, and therefore you can actually destroy your taste buds. But usually, unless you do really severe damage to your tongue, these cells will regenerate and will grow back. And then the other thing inside the taste bud is the afferent nerve. So first of all, the word afferent is going to come up a lot in the next few chapters, but basically it just means a sensory nerve. So afferent just means anything that's bringing information back to the brain, and usually that refers to a sensory system. So this nerve is a bundle of axons, and each axon synapses with a single taste receptor cell. And so this is the axon that's going to take the signal from the taste receptor cells back to the brain. So if we look at these receptor cells and look at how they respond to taste molecules, they respond to taste molecules with changes in membrane potential. Now, one thing to clarify, when we say taste receptor cell, what we mean is the cells that respond to taste molecules. The word receptor, though, can make things confusing because we've used that term before in the context of proteins on the membrane of the cell that respond to something outside the cell. And it means something similar here, but here we're not talking about a single protein, we're talking about an entire cell. So each receptor cell has the ability to respond to a taste molecule, or what we call a tastant. And so to figure out which tastant a taste receptor cell responds to, you can record the membrane potential of those cells, just like you can with neurons. So here we've got 3 different taste receptor cells inside a single taste bud, and there's an electrode in each one. These are electrodes that are stuck into the taste cells. Again, you would do this under a microscope, and you're recording the membrane potential of each cell. Sometimes, in fact, this is called the receptor potential, and that's just a complicated way of saying the membrane potential of a sensory receptor cell. So these graphs over here represent the receptor potentials of each of these receptor cells. And again, what they do in this experiment is they're applying different tastants and seeing how each one responds. So for example, here they're using sodium chloride as the tastant. Again, sodium we usually associate with the sense of saltiness. And in this experiment, what they're showing is that this cell here, what they're calling cell 1, is depolarized. So the membrane potential becomes more positive when sodium is applied. Same with the cell 2 here. So it also becomes depolarized. Whereas if you apply quinine, quinine again is the prototypical bitter tastant, and this cell 1 does not respond to quinine at all, meaning it does not change its membrane potential when you apply quinine. Meanwhile, quinine applied to cell 2 does create a small change in membrane potential, small depolarization. And then here we've added HCl. HCl is hydrochloric acid. Again, chloride doesn't do much, but if you add hydrochloric acid to water, it dissociates into chloride ions and hydrogen ions or protons. And those protons are going to lower the pH. So this should activate any sour receptors on the tongue. And in fact, what we see is this cell has a small depolarization in response to HCl, and this one has a larger depolarization. Finally, we've added sucrose. Sucrose is the tastant associated with the sweet sensation. And in this case, cell 1 and cell 2 don't respond at all. So they don't have any depolarization in response to sucrose, but cell 3 does respond. So that depolarization is how the taste bud cell, the taste receptor cell, responds to the tastant. And we know that because if you can also record from these afferent axons. So these are the axons that synapse with these taste receptor cells, and you can also record from those or from the nerve that they're connected to. And so here are the electrodes in those axons. And so, for example, cell 1 here is synapsed with axon 1. So these two are connected. And so you can see that when cell 1 is depolarized, so here and here, you can record the action potentials from that axon. And so each of these horizontal lines is an action potential. What you can see is that axon 1 fires a burst of action potentials in response to depolarization of cell 1. Meanwhile, axon 2 does the same thing. So when axon 2 fires a burst of action potentials, it's whenever cell 2 is depolarized. And the amount or the number of action potentials per second depends on the depolarization of the receptor. So when you apply quinine to cell 1, basically nothing happens. In other words, it fires, so we're still seeing action potentials, but the firing rate is much, much less. So the number of action potentials per second is much slower when you apply quinine than when you apply sodium chloride. Meanwhile, cell 2 does fire a burst of action potentials, but it's not nearly as intense as when you applied sodium chloride. So the way that the axon sort of encodes how strongly the taste receptor cell is activated is by changing its firing rate. And you can see in axon 3, or cell 3 doesn't respond to basically anything except for sucrose. So whenever you apply sodium chloride or quinine or HCl, the firing rate of axon 3 stays the same. So only one action potential, every few seconds or so. But then when you apply sucrose, suddenly it really responds. So you see a burst of action potentials only when sucrose is applied. So this is how each of these afferent axons is going to go to the brain and convey that signal. So that burst of action potentials from axon 3, for example, would be the signal to the brain that cell at least was exposed to sucrose. And so this kind of summarizes, this graph over here summarizes everything. And you can see that each individual receptor kind of encodes a different set of tastants according to a specific pattern. So the question is, though, how does the taste cell actually detect and react to the taste molecule? And so the process, and this goes for all the sensory systems, the process of turning a taste or a stimulus of any kind into a signal that your brain can interpret, which is an action potential, is called transduction. So all of the sensory systems have a transduction mechanism associated with it. So for taste, that means turning the presence of a tastant, a taste molecule, into a change in membrane potential, which then gets turned into an action potential. So again, the tastant is the original signal. The tastant has to cause some sort of change in membrane potential. And different tastants do this in different ways, which we'll talk about in a second. But in any case, or in all of these cases, changing the membrane potential or lowering the membrane potential triggers an increase in intracellular calcium. And that either means opening calcium channels in the membrane, so voltage-gated calcium channels, or releasing intracellular calcium stores. So that can mean calcium stores from the endoplasmic reticulum, for example. And then releasing calcium into the cell does kind of the same thing it does in neurons. So if you're at a presynaptic terminal, you should remember that the thing that triggers neurotransmitter release in a neuron is an influx of calcium ions. And so that's what happens at these taste receptors too. Calcium comes in, and that actually triggers the release of neurotransmitter from these cells. So these cells have synaptic vesicles, and they're typically releasing serotonin. Actually, these are usually serotonin releasing cells, and so they release serotonin into a synaptic cleft, just like a neuron would, onto the postsynaptic membrane of one of these gustatory afferent axons. So then there should be receptors on that axon that trigger an action potential when they're exposed to the neurotransmitter, and then the action potential should travel down the axon toward the brain. So that's how a tastant is transduced into an action potential. Now, and we'll come back and as we go through the different sensory systems too, we'll talk about other transduction mechanisms. So we'll talk about the transduction of sound, the transduction of light, and so on and so forth. But like I said, with taste, different tastants or different taste molecules are transduced in slightly different ways. So for salty and sour, again, the tastants in that case are sodium ions and protons, respectively. And because those are monovalent cations, both of them, they both have channels in the membrane associated with them that are selective, and so they both can enter the cell through those channels, and they just directly depolarize the membrane. So the sodium ion concentration inside of a taste cell is usually low compared to the outside, as with most cells. So when you eat food that has salt in it, sodium ions in it, then those sodium ions will accumulate in the saliva, and so the sodium ion concentration will go up in your saliva, and that will cause a greater sodium concentration gradient across the membrane. And we know from the Nernst equation that also will create an increase in the sodium equilibrium potential, which means that sodium will start to come into the cell in order to through sodium channels in order to get closer to equilibrium. And so that sodium coming in will depolarize the membrane. A positively charged sodium ion coming in will cause an increase in membrane polarization, or a more positive membrane. That depolarization travels from what we call the apex of the cell, where it's closer to the taste pore, down to the base of the cell, where you have voltage-gated sodium channels and voltage-gated calcium channels. So in fact, sodium coming in increases by opening voltage-gated sodium channels, increases the permeability to sodium, meaning even more sodium can come in, and you have an even greater depolarization. But the increase or the influx of calcium, which again is voltage gated, is what is important, because that will trigger the fusion of the synaptic vesicles and release neurotransmitter. Protons are a little more complicated because they also actually go through sodium channels. So these channels that let sodium through are actually not that picky. They'll let pretty much any monovalent cation go through. So protons can also enter. Again, protons have a positive charge just like sodium ions, so when they enter the cell, they cause the cell membrane to be less negative, or the membrane depolarizes. And again, that depolarization travels down the cell membrane to the base where it can trigger calcium influx. Now, the question then becomes, how does a cell, if both sodium ions and protons activate or go through the same channels, how does a salty taste cell, tell the difference between a sodium ion and a proton? And it turns out that these protons also interact with potassium channels on the taste cells. So if you have a proton coming into the cell, it's going to also actually inhibit these potassium channels. So the assumption is that the salty taste cells do not have these proton-sensitive potassium channels. So the sodium ions by themselves are enough to activate those cells. And meanwhile, protons entering the cell might not, the concentration difference might not be enough to trigger a response from the cell. Meanwhile, the sour taste cells have both the sodium channels and the potassium channels that are sensitive to protons. And so the protons coming in cause a depolarization, but closing off potassium channels, remember potassium usually has a higher internal concentration, so if you close potassium channels, you make it easier essentially for the protons to depolarize the membrane. So those two working together cause the depolarization. Now, for bitter, sweet, and umami, things get more complicated still, because you can have ion channels that will just let either sodium ions or protons come in, because those are small single atom ions, and so as long as there's a concentration gradient and a membrane potential, they have an ionic driving force to bring them in. But with bitter, sweet, and umami, all three of those sensations are associated with bigger molecules. So, bitter, again, things like quinine. Sweet is associated with things like sucrose. Umami associated with things like glutamate. And those molecules are too big to really go through a channel. So you can't have a sucrose channel, for example, because sucrose is just too big of a molecule. So having any channel that would be big enough to let sucrose in would let a bunch of other stuff in. So instead, all three of these sensations are associated with G protein coupled receptors. So we've talked all about G protein coupled receptors before in the context of synaptic transmission. And they work basically the same here. So you have a receptor in the membrane, which is coupled to a G protein, which is inside the cell. And so each of those flavors, each of those types of tastins has a different type of receptor associated with it. But all three of them work through basically the same mechanism. They activate the second messenger, or the G protein activates an enzyme called phospholipase C. Phospholipase C converts a lipid molecule called phosphatidylinositol bisphosphate, PIP2, into another molecule called IP3, or inositol triphosphate. And that has the effect of triggering the release of intracellular calcium stores. So calcium gets released from the smooth endoplasmic reticulum in response to IP3, and that calcium then has the ability to actually, and then you also have IP3 indirectly causing depolarization, and in any case, the calcium then has the ability to trigger synaptic vesicle fusion. There are actually a whole bunch of bitter receptors. So there are about 30 different bitter receptor proteins, and they're all called the T2R proteins. Whereas the sweet receptors, they're actually a complex of two different proteins that are stuck together. So for sweet receptors, you have the T1R2, the T1R3. So those two together detect molecules like sugar, sucrose. And then umami is associated with a different complex of receptors, T1R1 and T1R3. So actually T1R3, you can see, is associated with both sweet and umami, but it's the combination of receptors that really determines what the receptor responds to. So, and each receptor cell typically only expresses one type of receptor protein, but sometimes you can have cells that express more than one. Now, I said that taste is one of the simpler sensory systems because there's only the 5 sensations, but it turns out it gets a little more complicated than that because there are actually at least 3 kinds of taste receptor cells that have different combinations of receptor proteins, and which type of taste they detect again depends on the combination of receptor proteins they express. But this picture here, you don't need to memorize, but I just want to point out that the taste associated with a taste cell can become kind of complicated because it turns out it's not just about the receptors they express. There's also a lot of kind of crosstalks. The taste cells kind of communicate with each other, and so the actual signal that gets sent to the brain can be a complex combination of different signals or interactions between receptor cells. So once a taste receptor is activated, it sends a signal to the brain through what's called the central taste pathway. So this is how all sensory systems work. There's a sensory receptor somewhere in the body, and that sends information along a nerve to the central nervous system, either directly to the brain through one of the cranial nerves or through the spinal cord. In the case of the sensory systems, all the gustatory information, in the case of the taste sensory system, all the information comes along 3 cranial nerves, cranial nerve 7, cranial nerve 9, and for the back of the tongue, or what's called the epiglottis, cranial nerve 10, which is also called the vagus nerve. And then those axons synapse onto neurons in the medulla, in a region of the medulla called the gustatory nucleus. So these are, first I should say, these cranial nerve sensory neurons, their cell bodies are usually kind of out here in the right here where they get close to the brainstem, but they have a central and a peripheral process, both of which are axons. One that the peripheral process is an axon that brings information in from the tongue, and then the central process is an axon that goes into the brain and synapses onto, again, the neurons of like I said, the gustatory nucleus. So this is, again, these are the afferents from the tongue and epiglottis. The cell bodies of these neurons sit outside the brainstem in what are called the cranial nerve ganglia. And then they have their central process comes in and synapses onto neurons in the gustatory nucleus. So this is in the medulla, so we're right here in the brainstem, and the gustatory nucleus kind of sits on the dorsal side of the medulla, and there's one for each side. So there's a left gustatory nucleus and a right gustatory nucleus, and then those neurons, their axons, after being stimulated by the afferents from the tongue, their axons go up into the brain and synapse onto neurons in the thalamus. So now we're up in this region of the brain now. Remember, the thalamus is part of the diencephalon. So the gustatory nucleus axons synapse onto neurons in a region of the thalamus called the ventral posterior medial nucleus, or just the VPM. This is kind of how the thalamus is laid out. It's got a bunch of different nuclei. Each nucleus is usually named after roughly where it's located. So this nucleus is in the ventral posterior medial part of the thalamus, so that's where the name comes from, just the VPM. And then those neurons have axons that come out and go to the cerebral cortex, specifically to what's called the primary gustatory cortex. So this is the part of the cortex that gets, by definition, gets its input from the VPM and therefore processes gustatory information. And in fact, every sensory system, as we will learn, has its own primary cortical area. So this region, which corresponds to The primary gustatory cortex happens to be in the inferior parietal lobe. So this region here is the parietal lobe. So this would be the inferior parietal lobe. And then a region called the insular cortex, which is this region here. The insular cortex is kind of tucked in between the temporal lobe. This is the temporal lobe and the parietal lobe. And so to see it, you would have to kind of pull away the temporal lobe. So that's why it's called the insula or insular cortex. But that's where the gustatory region is found. One thing that makes the gustatory system a little bit unusual is that it stays on one side. So all of the axons from the left side of the tongue and the epiglottis go to the left gustatory nucleus. Axons from the left gustatory nucleus go to the left side of the thalamus, the left VPM, and then the axons from the left VPM go to the left primary gustatory cortex. So everything stays on one side. As we'll see, a lot of the other sensory systems cross to some extent. So input from the left side goes to the right side. But that's the pathway that information takes from the tongue to the brain. And generally speaking, when the information gets to the primary gustatory cortex, so there are going to be neurons here in primary gustatory cortex that are synapsing with those neurons from the thalamus. Those are the neurons that, once activated, where you have the conscious perception of taste. So in fact, that's an important thing about the sensory systems, is that the actual conscious awareness of a sensation typically happens in starting with the primary cortical area. Now there can be, and we'll see later, there are other sensory systems that have secondary, tertiary, even fourth degree, fifth degree levels of cortical areas that are linked to them. The gustatory system is a little bit more straightforward. There are other parts of the cortex that respond to gustatory input, but the primary gustatory cortex and the primary sensory regions for all of the sensory systems are kind of the lowest level at which you usually detect conscious awareness of a sensation. Which means, of course, you can directly activate these neurons and elicit taste sensation. So, in other words, if you had an electrode sticking in the brain here, and you activated it, then the patient would detect a taste. Now, what kind of taste they detect would depend on which population of neurons you activate. But that, again, is where conscious perception occurs. In fact, this is sometimes done. So with people that are getting brain surgery, sometimes the neurosurgeon will electrically stimulate the part of the brain they're working on to figure out exactly which region they are in. And again, if they do this, and they often do this on people who are awake, so typically when you have brain surgery, you are conscious. So they use general anesthetic for the skin and the skull where they need to drill in, but then you're kept awake for the procedures so that they can again stimulate these regions and ask you what you feel or what you sense, so that they know which region they're cutting into. Now, the last thing to say about taste, and this kind of applies to all the sensory systems too, is that the job of the sensory system is not just to detect a sensation, but to encode it. In other words, send all the information about that stimulus to the brain. And so we talked about the different parameters or properties of a stimulus that a sensory system has to encode. And so each sensory system has a different way of encoding the different properties of a stimulus. So with taste, of course, probably the most important thing to encode is the modality, meaning the type of stimulus that is appearing to the tongue. So which flavor or which taste actually is the tongue detecting. Now, the problem is that for each taste receptor can often detect more than one stimulus. And so you can see that here. This is a typical profile of a taste receptor. So cell 2, for example, responds to at least 3 different taste molecules. It responds to sodium, sodium ions. It responds a little bit to quinine, and it also responds to HCl. And we know, at least with sodium and protons, that they both go through the same receptor. So it's not surprising that the same cell might respond to both. Meanwhile, this particular cell, at least apparently, also has bitter receptors. So, so all three tastins activate the same cell. However, each receptor cell only activates one axon in the case of the gustatory system. So, so that's one thing that makes it simpler, is that each axon only responds to one taste receptor cell, even though each receptor cell activates or is activated by more than one stimulus. Now, in some cases, some of these cells, like cell 3, for example, only responds to sucrose. So cell 3 only cares about sucrose. Nothing else seems to really activate it. So cell 3 and the axon it connects to, axon 3, could be an example of what's called labeled line encoding. And this is the most straightforward way of encoding the identity of a stimulus. Basically, if you have a receptor that only responds to one stimulus, and that only activates one axon, then that axon essentially becomes a labeled line. In other words, the brain just knows or always can respond to a signal from that axon 3 in this case, the same way, because it's always going to encode the presence, in this case, of sucrose. So some sensory systems can use labeled lines to encode the nature or the type of stimulus that is present. But of course, that doesn't work if you have cells like cell 1 or cell 2 that respond to multiple stimuli. And of course, the other problem with labeled line systems is that you can only have as many different stimuli as you have lines. So if that might be possible in the gustatory system, where there's only 5 different stimuli that we respond to. But in most of the other sensory systems, as we will see, there are many other types of stimuli. And so the labeled line system becomes kind of unwieldy. So instead of a labeled line, what happens in not just in all the sensory systems, including taste, is you have what's called population coding. So instead of each stimulus being encoded by a single type of receptor or a single type of axon, any given stimulus can activate a number of different receptors, and sometimes it's a small number, sometimes it could be a large number. But any given cell can be activated by more than one stimulus. But generally, each stimulus always activates a specific population of neurons. So over here, for example, each of these little graphs just shows for a bunch of different neurons. We've got 40 different neurons here, how they respond to different tastins. And you can see that each neuron kind of has its own unique profile. But when you add them together, you see that there are populations. So for example, this population here really prefers sucrose. So that population, if you applied sucrose to the tongue, those cells would all kind of respond together and tell the brain that sucrose is present on the tongue, because they don't really respond to other cells. So those cells could, again, be used perhaps as a labeled line system. But when you look at sodium chloride and hydrochloric acid, again, some cells that respond to sodium chloride also respond to HCl, whereas some only respond to HCl and not chloride. There don't seem to be very many cells that respond specifically to HCl and not sodium chloride. Maybe this group down here. So again, if the stimulus is sodium, as salt, then these cells all firing would tell the brain that the stimulus is salt, and the sensation of saltiness would be perceived. Whereas if, say, this population of cells is active, these guys here, so like roughly 10 through 15, and then these cells down here, which correspond to roughly 33 through 35 or so, those cells, those populations together would tell the brain that there's hydrochloric acid present, and it would be perceived as sour. And meanwhile, this tiny population These are the only ones that really respond to quinine. But as long as only those cells are active by themselves, that would be perceived by the brain as the bitter sensation. So that's the point. You have 4 different populations of cells, each giving its own kind of unique response to different stimuli, and they can be overlapping. So the population that responds to hydrochloric acid, for example, overlaps with the population that responds to sodium chloride. But as long as they're not completely overlapping, then the brain can interpret those separately. So that's what we mean by population coding, and we'll see examples of population coding in the other sensory systems also. And so next time we will talk about the sense of smell.